are now on the unit 4. For the nursery practices, we have our first topic is seed germination. So what is seeds? So it is the basic input for vegetable production. Its quality is a major factor which determines the economic success of the crops by Mayer in 1982. Seed, seed germination, okay, take a look at the picture. So in plants, it is the process by which a dormant seed begins to sprout and grow into a seedling under the right growing condition. Dormant means resting stage in which when you are disturbed, when the is being disturbed, so it will grow and begins to sprout. So resumption of growth of the plant embryo following dormancy. So for the germination, it is defined as the emergence and development from the seed embryo of those essential structures. First, environmental conditions must trigger the seed to grow. So when water is plentiful, the seeds fill with water in a process called imbibition, so absorbing of water. So the water activates special proteins called the enzymes. So the leaves continue to grow towards the light source in process called photomorphogenesis. So wala yung R, no? Photomorphogenesis. So the structure, as a broad generalization, every seed consists of three essential parts. So an embryo, which will continue course, give rise to the new plant. So embryo also holds the nutrients and the food of the seeds. So it acts as a storage. So storage tissues which contain the substance which will nourish the embryo during its development prior to and for some time after germination. And another protective covering which is called also a testa or the seed coat, which shields the embryo and endosperm and may also play an important part controlling factors which initiate germination of seed, in particular entry of moisture and gaseous exchange. So we have now the pictures. We have external and internal part of the seeds. So for the external, we have seed coat or the testa, which is the covering of the seeds, or the hilum, and or another term for hilum. And for the embryo, we have cotyledon, the epicotyl, the hypocotyle, the plumule, and the radical, or the radicle, R radicle. Okay. For the common bean, we have now the seed coat, which is the testa, the plumule, the radicle, which radicle will grow first and will sprout first into the seeds and will meet the soil to get some nutrients in the soil and absorbs water as well. So the parts of the seeds are, first is the embryo. It consists of two important parts, which is the radicle and the plumule. Second is the radicle. It is the first organ to appear when a seed germinates. It's downward into soil, anchoring the seedling. So third is plumule. It is a part of a seed embryo that develops into the shoot bearing the first true leaves of a plant. So it will create the true pair of leaves. As you can see, when you try to germinate seeds. So it is often considered when the seed is ripe. And all that is necessary to promote germination is the right combination of moisture, temperature, and light, perhaps following interval to allow for after ripening. So fourth is the endosperm. The endosperm provides foods for the development of the seedling. So when endosperms has already done and had no food, so the radicle will now sprout and watch and gather nutrients from the soil or from the environment. And fifth is the seed coat. The seed coat consists of one several layers of different constitutions. This structure or combination of structures function not only as a mechanical protection for the seed, but is also important in many cases as a germinating regulator by controlling moisture and gas exchange, so between the seeds and the environment. So when the seed coat is tighter or more thicker, so it needs a process that will enhance and easily the seeds will easily imbibes water for them to germinate. So requirements for germination. First is substratum. It is suitable substrata for seed germination include paper, toweling, the blotter paper, the filter paper, the cotton, the sand, or the soil. So it is another term for stratification in which you're going to filter paper or or sand to make it rough side and having some pores of the seeds to make it easily to imbibe water. So imbibing is absorbing of water. Second is moisture. The moisture content is the amount of water in the seed and usually expressed as percentage. So seeds are kept damp by keeping them in chambers where relative humidity is maintained at 19 to 95%. But then some of the seeds, which is examples of orthodox seeds like rice and corn, yeah, it's an example, but then it must be stored in a lower percentage of moisture than to the recalcitrant seeds like durian and citrus, in which it needs some higher content of water to germinate. So third is temperature. So the seeds of most agricultural plants germinate somewhere in the temperature range of 10 degrees Celsius to 35 degrees Celsius. So temperature control is useful where dormancy occurs. So in dormant seeds with permeable seed coats, there seems to be certain blocks to the germination which may be surrounded or bypassed by various means. Some of the methods used are first, heating seeds at 40 degrees Celsius or higher temperatures. Second, using sharp alternation of temperature. Third, applying a period of moist or low temperature, which is vernalization. And second is for the heat treatment. That's the thing. And fourth is providing light. So fifth is treating with chemicals such as potassium nitrate or deuteria. Next is the light. So a few seeds prefer to germinate in darkness. Like when you germinate mung bean, it will grow lighter stems, but then um, lighter color of stems like white to green. Then so fully sun-raised germinated seedlings. So that's the difference. The color and the length of the seedlings. So seeds which require light should be grown on tanks, in petri dish, or in shade houses out of doors. Daylight or cool fluorescent light are preferable to tungsten lamps. So next is air. So a few water inhibiting plants germinate best under full moon or partly anaerobic condition. So however, most seeds require some aeration so that the test should not be kept too wet. So the process of seed germination, we have imbibition or the absorption of water by the seeds. So first step in seed germination is the absorption of water by the seed. So imbibition result in swelling of the seed as the cellular constituents get rehydrated. So second is respiration. It is the imbibition of water causes the resumption of metabolic activity in the rehydrated seeds. So we have aerobic and anaerobic respiration. So anaerobic respiration is mentioned earlier. So initially, the respiration may be anaerobic as oxygen begins entering the seeds. So for the third, effects of light on seed germination. Plants vary greatly in response to light with respect to seed germination. The seeds which respond to light for their germination are named as photoblastic. So for the germination, it is the breaking of dormancy. So the growth of the embryo and its penetration of the seed coat. So we have now the breakdown of barriers or the abrasion of seed coat, decomposition of seed coat with the help of the soil microbes, the gut enzymes, and cracking of seed coats, which is the use of the fire. Next will be the change in physical state, which is it will undergo rehydration. So next is destruction and dilution of inhibitors, the light, the temperature in the water. And next will be the production of growth promoters. So the processes behind germination, we have stages and the events. Okay, we have three stages: the pre-germination, the germination, and the post-germination. So we have now pre-germination, which have events, 6 events, and germination, which have 2 events, and post-germination has 3 events. So first, for the pre-germination, rehydration or the imbibition of water. So RNA and protein synthesis is stimulated and increased metabolism, increased respiration. So hydrolysis, which is the digestion of food reserves by the enzymes. So changes in cell ultrastructure, introduction of cell division and cell growth. For the germination, during this germination, so it is the rupture of the seed coat because radicle will soon to arise. A radicle will go out easily. And emergence of seedling, usually radicle first, as what I said. So for post germination, we have 3. So first is controlled growth of root and shoot axis. B is controlled transport of materials from food stores to growing axis. And last is the senescence, which is the death stage or the dying stage of a plant, or the aging for another term for germination. So a food storage tissues. So for the process of seed germination, again we have imbibition, as I mentioned earlier, with the same parts, and with the help of gibberellic acid, so which is a plant hormone similar to steroids, which dissolved and distributed by water. So for the gibberellic acid arrives at Allirin cells and activates certain genes. Then for the transcription, we have transportation, translation, and amylase. And the fourth is amylase accelerates hydrolysis of the starch. So we have seed germination, hydrated starch moves to the cotyledon and radicle to initiate growth. So it will help to push the radicle to go out. Therefore, it will see and how the world works, and also radicle will also the first to absorb water for it to grow. So this is the process of seed germination. For the monocot seed germination, we have here like coconut, no? Example lang. But then another monocot seed germinations. mostly in vegetables, and have some in fruits. So the fruit plus seed coat. Next is the endosperm. Next is the embryo, the shoot apex, the radical apex, the cotyledon, the monocot, the imbibition, the water, the starch, the hydrolysis, the sugar, and the alluron layer, the storage protein. Hydrolysis with the help of amino acids and help the DNA to transcript RNA translation of the amylase, the exocytosis, the maltose, the starch, and the sugar, and push through over for the growth and another development. So for the seed germination, like mango, so embryo, the radical apex or the apex, cotyledons, the seed coat, the same parts like monocot, but then have different shapes. So hydrolysis, the sugar, and the growth. So you see, we have now here the photochrome photo reversibility, which is the PFR 60 nanometer and 730 nanometer. So steps in germination process, we have seeds absorb water, which is imbibing of water. Water cracks the seed coat, activating the growth. So the seed coat or the testa will now be cracking. So root shoot or the hypocotyl, again, the root shoot, which is called as hypocotyl, and begins to grow downward. For the stem shoot or the epicotyl begins to grow upward. So normally, growth continues and green leaves begin to develop. Okay. First picture is the hypocotyl and the last picture is the epicotyl. So for the germination process, we have a seed with a tiny embryo inside remains in the ground until conditions are right for the seed to germinate. So after the seed coat splits, the embryo root begins to grow into the soil. Now the embryo can get water. Food in the seeds feeds the embryo as the schools or the stems grow upward the light. So the stem grows, leaves develop, and roots spread in the soil. And now the seedling can make its own food. So that's the process of germination. So steps for seed germination. Types of germination. We have epigeal germination, which is in this type of germination, the cotyledon are raised out of the soil and generally become green and photosynthetic. In dicots, they are pushed up by rapid extension of hypocotyl before growth of the epicotyl. So occurs in bean, the castor, the mustard, the tamarind, and sunflower. This is the epigeal germination in which the embryo, the cotyledon, the plant carries the cotyledon upward. So for the hypogeal germination, we have the cotyledons remain underground. Hypocotyl growth is restricted. The epicotyl grows towards raise the first leaves out of the show. Occurs in dicotyledonous seeds of gram, pea, mango, ground, and others. So monocotyledon like rice, maize, wheat, and others. So this is the example of hypogeal germination in which the embryo lives and in the ground as always. Unlike epigeal disease. So so the special type of germination, we have Viviparous. Okay, for the third is the Viviparous germination. So this special type of germination occurring in mangrove plants. So these plants generally grow in salty lakes, sea coast, and deltas. And they began to grow like in salty. But then, mangroves can be seen at Surigao, Surigao del Sur, and Surigao del Norte when so here the seed germinates while still attached to the parent plant. So the embryo emerges out of the fruit with a massive radicle pointing downwards. So due to increase in weight, the seedling separates from the parent plant and establishes itself in the muddy soil below. Example is Rhizophora. So this is the examples of vivipary in which the seeds will automatically matanggal siya. So viviparous seed germination in mangroves. Okay, that's all for this day. Good morning, good day, and thank you.